Hi everyone, my name is Aminata Coulibaly and I'm an assistant professor here at WVU in the Department of Neuroscience. Welcome to your graduate program and welcome to WVU. In my lab, we are interested in understanding how the immune system and the central nervous system interact. Specifically, I'm interested in how the activity of innate immune cells contributes to cognition. Now, our definition of cognition is an animal's ability to learn and adapt to changes to its environment. In terms of immune cells, some of you may know this, some of you may not, but there are two branches of the immune system. You have the innate branch and the adaptive branch. The innate branch is the one that's usually the first to be activated whenever an organism is challenged, either with a pathogen or through some type of injury. Within this compartment, within the innate immune cell compartment, I'm specifically interested in neutrophils. Now, neutrophils are the foot soldiers of the immune system. They're the most abundant cell type, and they're the ones to be activated whenever any type of perturbation occurs. In the central nervous system, neutrophil activation usually occurs super early after the injury or after pathogenic challenge. Now, my interest and the interest of my lab, the goal of my lab is to understand how the activation of neutrophils in the periphery, that is the rest of the body, or in the central nervous system or near the central nervous system, how does this activity within these compartments affect cognition or the animal's ability to learn and adapt to changes to the environment. And to do this, we use two different paradigms. We look at the animals in health, after injury, and in aging. For the health system, in a healthy organism, we look at how activity of neutrophils affect learning and memory and higher cognition function, so their ability to learn and unlearn tasks quickly. In terms of injury, we focus on stroke and in subarachnoid hemorrhage. And the subarachnoid hemorrhage is just a bleeding that occurs in the outer covering of the brain, and the other covering is called the meninges. In terms of aging, we want to know if activation of neutrophils affect normal aging processes, or if it contributes to the progression of the Alzheimer's disease pathology or exacerbate the condition of that neurodegeneration in response after neutrophils are activated in different compartments. And to look at these questions and to address hypotheses that we have going in the lab, we use a mixture of behavioral analysis, molecular analysis, as well as imaging. In terms of behavioral analysis, because we are interested in cognition, learning, and memory and such, we do a lot of behavioral tests in the lab. We are very fortunate here at WVU to have the behavioral core, which is really instrumental in getting those type of assays done and analyzed. In terms of molecular techniques, we use your basic benchtop assays, looking like ELISAs to look at protein levels, Western blotting. We may also do some PCRs and RT-PCRs to look at transcript level to see which genes are changing after our manipulations. And in terms of imaging, we use most often confocal imaging and fluorescence microscopy. We are looking to move into intravital imaging, which will be looking at how cells are trafficking change in the body, specifically neutrophils, when mice are undergoing specific cognitive tasks. So if you have any questions or you want to learn more about the work in my lab, please feel free to contact me.